Hello everyone and welcome to today's webinar, The Essential Role of MicroRNA and mRNA on Cancer Progression and Biomarker Discovery, presented by Dr. Malte Buchholz, Head of Basic Research, Clinic for Gastroenterology, University of Marburg. My name is Vidya Venkatesh. I'm from Thermo Fisher Scientific and I will be your moderator for today's event. We are delighted to bring to you this educational webinar presented by LabRoots, sponsored by Thermo Fisher Scientific. Before we begin, I would like to remind everyone that this is an interactive event. We'd like you to ask as many questions as you like during the presentation as well. Just click on the green Q&A button located on the lower left of the presentation window and type your question in the box that appears on the screen. We will answer as many questions as we have time for at the end, and we'll get back to you for those that we didn't get to during the live event. Please note that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. The presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. I'd now like to introduce our presenter. Dr. Malte Buchholz obtained his PhD in molecular biology from the University of Göttingen, Germany in 1998. He's group leader and head of basic research at the Department of Gastroenterology, Endocrinology and Metabolism, University of Marburg Hospital, Marburg. For more information about our speaker, please do click on his picture at the top left corner. Without further ado, I hand it over to Dr. Malte Buchholz to take us through the informative presentation.
Thank you, Vidya, for the kind invitation to present some of our data here. And I'm happy to do that, and I'd like to start off with just a couple of seconds explaining where we're coming from. As the title of this talk already hints at, we are working on pancreatic cancer. And pancreatic cancer, if you are hearing talks about it, they are often started with the sentence that pancreatic cancer really has the worst prognosis among all solid tumors. So what does that mean in practical terms? So this slide that I borrowed from the American Cancer Society shows a couple of numbers about survival rates of different tumors. So the percentages of patients that are still alive 5 years after they first got their cancer diagnosis. And if you look at the lower right, you'll see that, for example, prostate cancer has a very good prognosis. Almost 100% of patients are still alive 5 years after diagnosis. And right next to it, breast cancer has improved tremendously over the last years and decades, and almost 90% of the patients are still alive 5 years after their diagnosis. But if you now look for pancreatic cancer all the way up here on the left, you'll see that the number is only 5%, and in reality it's actually even lower. So less than 5% of patients will still be alive 5 years after they first were diagnosed with pancreatic cancer. And there's a couple of reasons for this dismal prognosis, and chief among them is the fact that the tumor is extremely resistant against chemotherapy and radiotherapy. And the only hope for cure actually that we can offer to the patients is the resection, the complete resection of the tumor at an early stage. But this is complicated very often by the fact that we usually catch the tumor too late. So by the time that the diagnosis is final and is fixed, only between 5 and 20% of the patients are really amenable for surgical resection with the intent of curing the patient. All others can only be treated palliatively. So the complete removal of the tumor, which surgeons call the R0 resection, is actually the only hope for cure that we have at the moment. But for this, the timely and accurate diagnosis of the tumor really is crucial. And what is usually done in the diagnostic workup of patients who are suspected of having a malignant tumor in the pancreas is that a fine needle is advanced into the tumor and some material is aspirated from this mass, and then it's expelled onto a microscope slide and then a pathologist tries to determine if there is evidence of malignant cells present in this cytology. And this works reasonably well when there is malignant cells present in the aspirate, but very often also the diagnosis cannot be made because the sample is non-diagnostic. And in very experienced centers, the accuracy of diagnosis of this procedure is around 80%. So our idea many years ago already was to improve this situation by including other forms of diagnosis. So recover residual material and do a molecular analysis of this material to get to a better and faster diagnosis. And what we did a couple of years ago was to create specialized cDNA arrays. And for that, we took our own data, but also gene expression data from other sources, from stage libraries, from the literature, from other biobanks or gene banks. And we assembled a set of a total of 588 features or genes that we assembled on these cDNA arrays. And then we tested if these arrays could give us diagnostic information if we use it to analyze finding the aspiration material from patients. And the case that we studied, the differential diagnosis that we looked at first, was the differentiation between malignant tumors, which is called PDAC, pancreatic ductal adenocarcinoma, and on the other hand, an inflammatory tumor in the pancreas. It's called chronic pancreatitis, which of course needs to be treated completely differently. And this turned out to work very well. So we analyzed a total of 62 tissue samples and divided this into a training set and a test set. We produced these gene expression profiles from these samples. We looked for principal components in these data and constructed from that a classifier, and then we tested this classifier on the test set of 20 samples that had not been used to train the classifier. And we saw that we got a very good diagnostic accuracy of 95% out of this, and this is represented by this little schematic on the right side of this slide. So this was encouraging, and it showed us that gene expression analysis from minute amounts of biopsy material from pancreatic tumors can really aid in the diagnostic decision, distinguishing between inflammatory tumors and malignant cancer. But the situation was not ideal in our view, because this system that we used, the cDNA arrays, is a very experimental system. It used radioactive labeling of the samples, very long exposure times to get the information for each individual patient sample. So we were looking for a system where we could use the same principle, but on a better technological basis. And we found it in the Techmen array system, microfluidic cards. And for those of you who may not be familiar with Techmen arrays, this is a schematic representation of what these arrays look like. So this here is one Techmen array card, and this here is a larger representation of what this card looks like. And as you can see, it has all these little blue reaction chambers within the card, and every reaction chamber is served by one of these loading ports here. So pipetting a sample into one of these loading ports will serve 48 different reaction chambers, and in each of these reaction chambers, one Techmen array will be pre-deposited so that a real-time PCR reaction will take place at this point for one specific gene. And what we thought would be advantages of this system was we had a selected set of genes from our previous analyses. Real-time PCR offers a much greater dynamic range than radioactive labeling of samples, more sensitive detection, of course fast analysis, no 5-day exposure times, and much easier handling because most of the components of the reactions are already deposited in the slide, and we only have to add the samples. The next aspect that we wanted to also include while we improved our diagnostic system was that we wanted to also include the analysis not only of mRNA markers but also microRNAs. And the reason behind this was that it has been known by the time already that microRNAs are important players in regulation of gene expression, that they are often misregulated in pancreatic cancer along with their target mRNAs, and that they are, because they are so small, very stable in all kinds of analytical material. So potentially very good marker molecules as well. So we thought that simultaneous detection of mRNA and microRNA would be highly desirable, and we started working together very closely with the folks at Applied Biosystems, which is now part of Thermo Fisher, and we tried to implement that in one single Techmen array card, microfluidic card. And this is what the initial version of such a mixed array looked at, like in our hands. So we selected 79 of our mRNA genes from our previous analyses, along with 6 mRNA reference genes that we used to as a reference for normalizing the data, the mRNA data separately from the microRNA data. And we selected 9 microRNA genes that had been described in the literature of being differentially expressed between pancreatic cancer and chronic pancreatitis, and we included 2 small RNA reference genes for normalization of the microRNAs. And together, this was a set of 96 genes. As you can see here, this was represented on 2 loading ports, or served by 2 loading ports in one of these microfluidic cards, so that each sample was loaded into 2 of those loading ports, which means that on each of these Techmen array cards we could analyze 4 different samples, 4 different patient samples. And the protocol that we used for the analyses of these samples back then was slightly different than what we're using today. We aspirate the tumor material, as I showed you a couple of slides earlier, did a total RNA extraction, mRNA and microRNA simultaneously from these samples. cDNA was prepared from these samples using custom gene-specific primer pools for the mRNAs and the microRNAs. And then we used the Techmen microRNA reverse transcription kit to produce cDNA from that, pre-amplified the whole mix. All this was done in one tube. Pre-amplified the whole mix using a custom gene-specific primer pool and limited cycling, and then loaded these mixed samples onto the Techmen arrays. And we did a couple of analyses first to ensure that this gave us reproducible data for both the mRNA and the microRNA genes. And I'm not going to go into too much detail on the analyses that we did, but we produced RNA pools from pancreatic cancer cell lines for this purpose. We worked up these RNA pools on different days with different replicates to make sure that we could compare how reproducible this procedure was from a purely technical standpoint. And the results were very good, as you can see here on this heat map, where we charted gene expression value, which is encoded as green for low expression, red for high expression, and gray for missing value. You can see that the technical replicates, they cluster most closely together, as shown here. And if you look at the same samples on 2 different days, this is the next level of replicability here, and you see that they're very, very similar. And if we have the RNA pools once unamplified and once pre-amplified, then these again cluster very closely together. if we compare this to material from primary tumors here on the right-hand side, which are clearly separate from the RNA pool replicates. So high level of reproducibility and appropriate detection and pre-amplification of both mRNAs and microRNAs. So that was very encouraging, and we went on to analyze a large series of prospectively collected fine aspiration biopsy samples. These were from all kinds of suspect masses in the pancreas, and they included a number of 41 ductal adenocarcinomas, and 33 of these cases where there was just a chronic inflammation present in the pancreas, but not a malignant tumor. And this was the distinction that we concentrated on first. And the first question that we asked was, how will microRNA markers, the 9 microRNA markers that we included specifically because they were described in the literature to be good discriminators of malignant tumors and inflammatory tumors in the pancreas, how well would they perform in trying to distinguish between these samples? And to get a very good and conservative estimate of this, we employed a method that is called 10 by 10-fold cross-validation. I'm not going to go into too much detail of this. Just the basic principle is, if you look at the whole data set, which is symbolized here by this bar, and if you imagine that these red samples are the tumor samples and the green samples are the chronic pancreatitis samples, or the proportion of samples that are really chronic pancreatitis, then this is divided into 10 equal sets, which you can see below in the next step. And then a classifier is trained with 9 of these subsets and then tested on the 10th. This is what's symbolized here. And then in the next step, the whole thing is repeated, just that a different set of samples is left out of the training and then tested on later, all the way until all 10 subsets have once been left out of the training and have been tested on, and all the while it is always counted how many of the diagnostic calls have been correct or have been false, and from that a diagnostic accuracy is calculated. And if you do this with just the 9 microRNA markers that we selected, then the best performing classifier, we tested all possible combinations of microRNAs within this set, and the best performing classifier reached an accuracy of 75%. This is symbolized here by the circle on the left side, which is not bad, but of course by far not good enough to really do diagnostics. So next we looked at the performance when we take microRNA markers as well as mRNA markers, and this is again the whole set of samples, mRNA markers and microRNA markers for the distinction between chronic pancreatitis. This is given here as CP, and the malignant samples, that is given here as PDAC. And it's already a very simple analysis, a hierarchical cluster analysis, as symbolized here by the heat map on the left side, shows you that the distinction was relatively good even with simple analyses. So you can readily see blocks of chronic pancreatitis samples, and they are separate from the blocks of ductal adenocarcinoma samples. So for a set of 88 different markers, it's not possible to exhaustively test all possible combinations, but what we did was we selected, or we did an analysis where we ranked all individual genes, mRNA and microRNA genes, for the individual ability to distinguish between PDAC and CP samples. And then we assembled more markers to build classifiers, starting with the best individual performing classifier first. And then we tested in using the same procedure, the 10 by 10-fold cross-validation that I showed you earlier, how well these combinations of markers performed in the distinction between chronic pancreatitis and ductal adenocarcinoma. And what turned out was that a collection of 77 microRNA and mRNA markers in a mixed combination had the absolute best performance in the 10 by 10-fold cross-validation, which is very rigorous, and it reached an accuracy of 93% for the distinction between these samples, and this was accompanied by a sensitivity of 91.7 and a specificity of 94.5%. And what you see here on the right side of this chart is that if you train the classifier with all samples and then retest on all samples, you get 100% distinction between the two classes. So very high diagnostic accuracy, and shows clearly that the simultaneous analysis of microRNA and mRNA is superior to just looking at microRNAs alone. So this is about to be published. It's in press in Oncotarget, and you can read about this in more detail in a couple of days when the paper is online. But still, this was not the end for us. We still have more plans because the analysis of fine needle aspiration biopsies is still a very invasive procedure. So a needle has to be inserted deep into the abdomen of the patients to get the aspiration biopsy samples. And ideally, we would like to be able to do diagnostics non-invasively, which means using blood as an analytical sample. And this is where we are currently. This is ongoing research. We have produced another version of a mixed Techmen array for this purpose specifically. We selected from our set of mRNA candidates where we know that they are helpful in the distinction between ductal adenocarcinoma and chronic pancreatitis. We selected from them a set of mRNA genes that we can regularly see in the serum of patients as well. And these are managed with 6 mRNA reference genes. And we did de novo a whole miRNA analysis of markers, microRNA markers that are present in the serum of ductal adenocarcinoma patients, and we selected from those a set of 37 potentially diagnostic microRNA markers, along with 5 microRNA reference genes. So if you do the math, you'll see that together this is again a 96 gene panel, which takes up 2 loading ports on a Techmen array card. So again, we can analyze 4 patient samples on one of these mixed Techmen array cards. The specific idea that we're using this version of a non-invasive diagnostic, potentially diagnostic Techmen array for now is to detect recurrence of pancreatic cancer and to detect it earlier than we can detect recurring tumor with the methods that we have at the moment. And the protocol that we use for analysis of diagnostic samples is slightly different now than what I've shown you before, because there is a new generation of TaqMan assays for microRNAs out now that are called TaqMan Advanced assays. And the principle here, if you concentrate on the right side of the schematic, is that the microRNAs are amended by a poly-A tailing reaction and a ligation of a 5' adapter, so that you end up with a universal, or with the possibility to use a set of universal primers to amplify all the microRNAs that are present in one sample. And again, in the same tube, we do the reverse transcription of the mRNA markers using standard random hexamer protocols, and amplify those then gene specifically with primers that are specific to each of the 49 marker genes that we have. All this happens together in one tube, and then we can load those samples again onto the Techmen array cards and get the quantification of all the markers simultaneously. And the reason that we're looking for tumor recurrence is that it's been clearly shown that patients who suffer from a relapse do much better if the relapsing tumor is caught early on, and then it can either be operated again or treated with chemotherapy. So we have founded a consortium within Europe with partners from Germany, Italy, and from Madrid in Spain. And what we are doing within this project is following up patients who had undergone R0 resection of a pancreatic tumor and collecting serum samples during the follow-up of these patients and archiving these serum samples until the time point when there's a clinically overt diagnosis of tumor recurrence. And then we split up the samples into samples that can go into the no evidence of disease group. So at least 12 months after a sample like this has been collected, there was no tumor recurrence. And on the other hand, the samples where a recurring tumor was already clearly present, and we will use the Techmen array cards to determine if our selection of markers can clearly distinguish between these two types of samples from the same patient. Of course, we are in the process of collecting these samples, so we don't have this data yet, but we do have some preliminary data that I'm going to show you in a couple of seconds. And I'd like to take this opportunity to show you a little bit of how you can analyze data like that using the tools that the Thermo Fisher Cloud has to offer. So this is a cloud space where you can upload all your data, and you can not only store it there, but you can also use some tools on this site to analyze them. And this is done by creating a new project. There's a button that you can press for that, and you can then import data files for this specific project. And in our case now, what we want to do is a relative quantitation study. So you press the RQ button, and then you will be presented with a couple of options of organizing your data and then also analyzing your data for the specific task. So the first thing is organizing your data, telling the system which ones of your samples belong to what kind of biogroup. And in our case, again, as you have already seen now a couple of times, is we have chronic pancreatitis samples on the one hand and ductal adenocarcinoma samples on the other hand. And once you've organized your data, you can take a look at what did your plate setup look like, is all of this correct, and review your data in the next step. And you can do this either target specific, so look at what did your individual markers or genes look like across the different samples. This is what you see here. But if you select a different option in a tab like this here, for example, you can also do this plate-wise or reaction-wise, if you will. So you can go through your data before you do any kind of analysis to see how did they perform from a pure technical standpoint. And then for the analysis itself, there's a couple of tools that the system provides. A very informative tool here, of course, is again the hierarchical cluster analysis, but highlighted here as well, for example, is an option to produce box plots to look at marker expression across biogroups or samples. So a lot of options that you can use. And if you look at the hierarchical clustering that the data of our 88 specific markers produced, what you can see here is that the clustering clearly puts the chronic pancreatitis samples separately and apart from the ductal adenocarcinoma samples. So there seems to be clearly a difference present in the markers that you can find in serum from patients that suffer from either inflammatory or malignant tumors of the pancreas. What you also see is that the expression patterns among the group of ductal adenocarcinoma patients varies very strongly, and this is something that is known. There's a high variability in expression patterns among individual tumors, and this is another reason why looking at many markers simultaneously is probably a way to get to better diagnosis of this disease. Just as a very quick outlook, one thing that we're also working on together with the folks at Thermo Fisher is to try and include yet another level of detection in the mixed Techmen arrays. So we'd also like to be able to look for gene-specific mutations, so mutations that are typical for ductal adenocarcinoma tumors in the same analysis steps, so also including these on the mixed Techmen arrays. And the technology that could provide for this is the CAST-PCR technology, so a allele-specific PCR that could be used to detect specific mutations. So if it turns out to be possible to combine all three levels of genomic analysis in one Techmen array card, we could not only look for expression levels of mRNA and microRNA marker genes, but at the same time also detect presence or absence of specific mutations, which should further improve the diagnostic accuracy of the calls that we're doing. So with that, I'd like to thank the large number of people that have contributed to these studies over the years. Very specifically, I'd like to thank Harita and Kathleen from Thermo Fisher in South San Francisco, whom we are working with very closely on these projects, and then also collaborators from Germany, all over Europe, the group in Marburg, with Thomas Geis, Eike Geimer, Christian Bauer, and last but definitely not least, the people in my group who are actually, of course, doing all the work. And with that, I'd like to thank you for your attention, and I'm happy to take any questions.
Thank you, Dr. Buchholz, for that wonderful presentation. We are now ready for questions and answers from the audience. Please remember to enter your question into the chat box on the lower left corner, and we will answer as many as possible during the course of this presentation. The first question is from Irene. Are mRNAs and microRNAs free in blood or associated with extracellular vesicles?
Well, we personally haven't checked for that in our samples, but if you look in the literature, both will be true. So there will be mRNAs and microRNAs that are just associated to proteins like organoid, so more or less freely floating in the bloodstream. But there are definitely also microvesicles and exosomes that are circulating. We assume that we're catching both with the methods that we use, but we have no way of discriminating or attaching percentages, how many of the markers that we see are either free-floating or exosome-bound. So my answer would be probably both, but we haven't discriminated between them.
Great. Another question from Irene. Do we have the same technology for castration-resistant prostate cancer?
Well, in principle, the technology would work the same. The selection of markers would, of course, be different, or would probably be different for other types of cancer. And we have so far concentrated on pancreatic cancer, so I can't give any indication of which marker combinations would be sensible to test for prostate cancer. But in principle, the technology should work the same.
Great. We have a question from Christy. Do you think the mixed TaqMan arrays could be suitable for screening the general population of early-stage pancreatic cancer?
Yes, that would be a very nice thing to do because, of course, catching the tumor early would be the ideal situation to help the patient as best we can. The problem here is with screening that we're fighting numbers. The incidence of the tumor in the general population is really low, which is, of course, a good thing because otherwise we would see more deaths by pancreatic cancer. But on the other hand, the reason that, or the fact that there are so few people who contract the disease, although they will probably all die from the disease, means that we would have to screen a lot of people before we find one who actually has a pancreatic tumor that has not been detected yet. And in the process, we would invariably produce many false positive calls. So we would be confronting patients or people who don't really have cancer falsely with the diagnosis of pancreatic cancer. And so this is on two hands, on the one hand from the economic side and on the other hand from the ethical side, not really feasible. So we will have to concentrate on high-risk groups like tumor recurrence in patients that already had the cancer.
Great. We have a question from Emmanuel. He says, thank you for the presentation. Do we have any experience profiling fecal microRNA as a disease biomarker? If so, how reliable is fecal microRNA in pancreatic cancer biomarker?
No, actually we have not tried that. We have briefly worked with urine, with patient urine, and there is, in principle, we can detect microRNAs in urine as well, but we have not followed that up in any greater study or any greater detail. We have not looked for fecal mRNA. I know that other groups are using protein detection in fecal matter, elastase and other markers to try and distinguish between inflammatory and malignant processes in the pancreas. But we haven't looked for mRNA and microRNA markers in fecal samples.
Great. We have a question from Deborah. How close do you think a blood-based assay is, and which technology company do you think is the closest to commercializing a blood-based test?
Okay, so I can't really speak to that with any kind of authority because so we are an academic group in the hospital, in the University Hospital of Marburg here. So we are really coming from the academic side. Definitely the issue of commercialization is a whole other beast, and I have little experience in this field. And actually, so I wouldn't be qualified to answer who will be first to market. I think, but I mean, how close are blood-based assays? There are already some on the market. So, and in principle, detecting analytes in blood has been used for decades now. Of course, proteins of different kinds have been detected, and there are groups that are also looking for combinations of detecting mutations or genomic DNA from circulating tumor cells as well as protein markers. So marker detection in blood is, of course, nothing new. The specific combination that we are using here, microRNAs and mRNAs, I'm not aware that anybody is in the process of commercializing something like that. But, of course, I can be wrong.
Great. We have a question from Jorge. Is there any genetic background that produces pancreatic cancer?
Yes, there is. Well, we'll have to distinguish between, on the one hand, sporadic pancreatic cancer, where there is no indication that there is a genetic basis. It's purely or presumably purely arising from somatic mutations that are acquired through life. And on the other hand, there is a number of families where there's clearly a significant enlarged or heightened frequency of pancreatic cancer occurrences. And we call this familial pancreatic cancer. It's a syndrome that's well described, but in very few cases, in very few families, have we actually detected the causative mutations. Probably in these families as well, it's probably a multi-genetic occurrence. There are very few cases where we can point to a mutation in BRCA2 that is probably causative, but in others it will be a combination that's not so easy to detect.
Great. We have a question on the data analysis. So how is the data from the combined assays normalized? It seems like the normalization of these data sets is a popular question that's coming in.
Right. We included, let's call them housekeeping genes separately on the one hand for the mRNA assays and on the other hand for the microRNA assays. So the mRNA CT values, so that's the raw data that you get from these analyses, these were normalized separately to mRNA reference genes, and the microRNA assays, the CT values that come from those are normalized to small molecule reference genes. So in the first versions of our mixed arrays, these were small nuclear RNAs, and in the newer version now, we have included 5 microRNAs that are reported to be very stably expressed across all kinds of different tissues and body fluids. And our results indicate that these are indeed very stable. So that's what we're using for normalization in this context. We are, on the other hand, we are also constantly analyzing our own data to try and see is there any better way for normalization or for workup of the raw data before we make diagnostic calls. But so far, the classical normalization to so-called housekeeping genes is what works best in our hands.
Great. Could you speak to other scenarios in which simultaneous detection of microRNA and mRNA could be useful in?
Yes, so I can think of several actually. We are working in the field of cancer, and cancer is a disease of that arises from mutations that ultimately cause differences in gene expression. And we know that microRNAs are important players in regulating expression of other genes. So of course, if you have a specific pathway, specific set of mRNA genes that you're interested in, and you have a number of microRNAs that you suspect of being involved in regulating these specific set of mRNA genes, of course it would make sense to have them combined on a microfluidic card to be able to analyze them simultaneously. So if you think that there is a coordinated change of expression of microRNAs and their target mRNAs, this would be a nice case to use mixed Techmen arrays, for example, so you can just have all your answers in one view. And I'm sure there are many other scenarios where it would be advantageous to have a view on microRNAs and mRNAs simultaneously. And all kinds of combinations are possible with these mixed arrays, of course.
Great. One more question, and this is actually our last question of the day. The heat map showing clustering of CP and PDAC shows very little difference in expression patterns. Is this significant enough to distinguish between the two sample types?
Good question. I would actually disagree slightly. So I would not say that there is very little difference in expression patterns. What we do see is that there is considerable variation in expression of these individual markers in individual patient samples. And that's actually something that we've learned along the way, and not only us, but many people. Also, if you look at the recent publications on large-scale genomic analyses in pancreatic cancer, there is a large degree of intertumoral heterogeneity, and I'm not quite sure how to quantify this, but the authors of a recent Nature paper have actually concluded that pancreatic cancer is the tumor with the highest degree of intertumoral heterogeneity. So it's expected that you get fluctuation of expression of individual markers across different samples from different cancer patients. And that's exactly why we need, well, we probably will never find a single marker or just a handful of markers that will reliably distinguish between pancreatic cancer and other diseases for all the patients. It will probably always be a more complicated set of signatures that we can find in individual patients, and that's why we are working very closely together with bioinformatics departments to find those more complicated expression patterns. But our preliminary data so far seem to indicate that we will be quite capable of distinguishing between tumor patients and controls. The question will be what diagnostic accuracy can we really reach? So how accurate can we really become?
Great. We have many more questions coming in, but maybe we'll take one last question before we stop. The heat map showing clustering. Sorry. How can we distinguish between exocrine and endocrine tumor using this method?
So that's also a good question, of course, for those participants who are not so familiar with the field of cancer in the pancreas. So in the pancreas, you have two types of cells, the exocrine cells and the endocrine cells, and both can give cancers. The exocrine cancers are the bad ones. The endocrine cancers are much less aggressive, actually. And they produce a clearly different pattern of marker expression compared to ductal adenocarcinoma. So it's not a problem to distinguish between endocrine and exocrine tumors using these markers, but it's not a very pressing diagnostic need at the moment anyway, because endocrine tumors can be diagnosed very well already with the methods that are already in the clinic. But when we do get the odd endocrine cancer sample in our analyses, we can always clearly see that they're different from the ductal adenocarcinomas.
Great. Thank you so much, Dr. Buchholz. This was a very informative presentation, and many thanks to the audience for the engagement. For those questions that we didn't get to during the course of this webinar, we will certainly respond back to you privately. I would also like to thank LabRoots for making today's webcast possible. Before we know, I wanted to let everyone know that today's webinar will be available for on-demand viewing through May 2018. You will receive an email from LabRoots letting you know when this webinar is available for replay. Please share this announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Bye-bye, and have a great rest of the day.